And while they're doing that, I'm wondering, it's extremely difficult with the lights sitting here right in my eyes to look out there. I'm just wondering whether we can lift the lights in the main auditorium any further. That's better. Now I can see. You like to sit down? Then you can go to help to a mic. Let me see. There's 3 on each side. I want to start the discussion by trying to link the 2 talks on chickpea and chickpea roots. Mahendra, you spoke about this collection of chickpeas that were screened for root characteristics. You showed them in a root root pots or root facility, but I believe that they were also screened in the field with a trench system. And there's 2 questions I have. One is the ones that Professor Sadiq looked at, are they the ones that were also looked at earlier under field conditions or under pot conditions, but particularly under field conditions? And is there some linkages there that we can learn from that? And then the other question that I have for you is that you suggested that there was and it's been mentioned previously, that there's a genetic hotspot. You didn't say whether these for drought tolerance, you didn't say whether the root characteristics that you had identified also tracked into that hotspot or whether it's a different area. I don't know who wants to go first. Maybe you should go first. No?
So,
just a moment.
Yeah.
Thank you, Neil. Yes, I looked at the data generated by Kasavagi and others, and some of those lines were having greater root length density and other parameters. Not necessarily they were all correlating, but they were on the higher end. For example, ICC 4958 and the other line Mahinder showed had.
So I will answer the second question because first question is already answered. So the root traits are also lodged in the QTL hotspot that we identified. Root length density, for example, root length density and root surface area were coming in this particular QTL hotspot region.
Good. Yeah. And what about yield?
Yeah. So we have QTLs for several yield and yield related traits. So we have 100 seed weight in this particular region, and this explains about 58% of phenotypic variation available for this trait.
Okay, good. Questions from the floor or other members? Maybe some of the panel members have got questions for other members. All right. Is there anyone at a microphone? Yes, someone way at the back. Sorry, I can't see who it is.
Hi, Adam Price. I want to ask a question for the first 2 speakers about aquaporins because I've been struggling with those for a few years. It relates to the feature Steve talked about. They form heterodimers, and maybe it's quite a simple question. To what extent does the fact that they form heterodimers make it difficult to study them using molecular biology, to study their function using molecular biology? And to what extent does that also complicate our ability to model what will happen if we modify individual genes?
It's a very good question. Basically, the heterotetramers that form with the PIP1, PIP2 interaction are involved in targeting to the membrane. That's been shown by Francois Chamont's work. But it also affects their sensitivity to calcium and pH, as well as we've shown their selectivity. This has also been shown for CO2 transport. So it really increases the complexity and makes it very difficult for us to actually model unless we know what the degree of interactions are between the different types of aquaporins. So I'm not sure that I didn't actually catch all your questions, so I'm not sure that I'm getting onto the right track here, but yeah, okay. It's a problem that the whole field is struggling with. I mean, I showed a lot of stuff about transcripts and not much about actual protein content in the membrane. That's also a problem because the isoforms are so similar, it's very difficult to actually get quantifiable levels of the different isoforms in the membrane unless you're using some fairly sophisticated mass spectrometry techniques. But it's surprising actually that transcripts do correlate with actual functional measures of water transport in many respects. But nevertheless, it'd be nice to see what the proteins are doing and also what the proteins are doing in terms of interactions. So it's quite a challenge, and we're trying to tackle that ourselves in the connection with the non-selective cation work, because if the 2,1 in the membrane is always as a heterodimer, then it's not going to work as an ion channel. But if it's in the membrane as a homotetramer, then it will probably function as a non-selective cation channel. So we have to ascertain that. Sorry if I went on to.
Tony Fisher.
Thank you, Neil. My question is to the last two speakers, but especially the last speaker, who seemed to be relying on yield per plant as a measure of performance under drought. Now, if those plants are not in a plot surrounded by like plants, it's practically meaningless.
Thanks for that. And it's not the yield we looked at. What we wanted to do was to actually introgress the traits. And once the traits are introgressed, we know those trait introgression lines which have better root system and a better water use efficiency, which have been combined. And the measurement was done in the germplasm from some other surrogate methods like the carbon isotopes and the root measurements by root structures. And after we developed that by marker-assisted trait introgression, one of the ways by which we can ascertain whether the trait have any relevance at all under the field condition was to now look for yield under the aerobic conditions in the field. That's the reason why we, at the end, we say there's a lot of other physiological data that we have. But then for want of time, I didn't give that information. But when we look at as a result of the trait introgression under the field conditions, whether or not the increase in productivity is there is what we want to see.
I just want to see yield per hectare, please.
Oh, sure. Sir, what we have done is these lines that we have got now are some of these which have been selected based on the yield per plant. But then right now, this material is being looked at in several locations, such as in our institute, as well as Tamil Nadu Agriculture University. My breeder colleagues are here in this hall now. And at the end of this season, I'm sure we will have that kind of information, the yield per, not per hectare, but for sure in terms of 10 meter square area.
There's a question at the back first, Roberto.
Yeah, I have a question that it's
just state your name. I can't see who you are.
My name is Jamil Khan. I'm from University of Agricultural Science, Bangalore. And my question is related to the aquaporins. I was reading the paper of Gazzilio who says that the H plus PPAs, not ATPase, it's H plus PPAs who have the function of sodium exclusion, deep root, and they are the homodimers. So does it relate to the answer of that aquaporins have the same property, what you discussed for the H plus PPS and aquaporins? Have they any relationship?
I'm not sure I caught your question. The echo is quite bad up here. So based on the sequences of PIP2-1 in Arabidopsis and comparing it with other aquaporins in Arabidopsis, we've come up with some motifs in the Arabidopsis PIP2-1 which we're using to scan other aquaporins in crop species. A model grass that we've looked at, Soteria, we identified a PIP in that from this sequence motif, and we showed it was a sort of lucky hit. The first time we tried it, it turned out to be a non-selective cation channel. So I think across many species, we're going to find these isolated examples of some aquaporins that are allowing some ion transport. I hope I'm on the right track here. But It's interesting as well if we step a little bit wider and look at humans. The Arabidopsis PIP2-1 shows more similarities to aquaporin 1 from humans than it does to other PIP aquaporins in Arabidopsis. And it's some of these motifs that we see in the aquaporin 1 that are linked to this non-selective cation transport. So it could be that there's a couple of these isoforms in each species that carry out this sort of interesting transport. So sorry if I'm off the track because I didn't actually hear much of your question.
Does that answer your question?
Oh, maybe we can discuss it later because it was hard.
Roberto.
Roberto Tuberosa, congratulations to all the speakers, and I think it shows the quick comment that roots comes of age, as we all say. The question is for Stefan, and being a QTL guy, you probably can read in my mind what my question would be. Although QTLs is not your cup of tea, if you look at the literature, what evidence is there, and if so, how solid it is in terms of relationship between aquaporins and grain yield under different water regimes?
Yeah, I think so. Well, I think there in rice, I think there's evidence that some aquaporins come in under QTLs because unfortunately I missed the talk in the workshop that linked that. But then, you know, there are many small traits, many traits that contribute to drought tolerance. I mean, the physiological evidence which suggests that aquaporins are probably important for water transport because that's mainly the substrate that they transport. But I suspect that the things that will come in under QTLs will be the controllers. So the kinases that phosphorylate them, the proteins that traffic them to the membrane. And there's all these other controllers that are actually affecting the expression of these guys. And I suspect that the QTLs might actually pull up those proteins more so than the actual individual aquaporins. But I'm not really familiar with any evidence that's brought up aquaporins under any QTL yet, but I believe there is some in rice, but I'm not sure of the data. Maybe
I was at that workshop and there was some in that direction, but I wanted to know if you had looked deeper in the literature to answer your question.
I'm not familiar with any others other than that report. But maybe...
No, it's okay. It's okay. Yeah. Thank you very much for your answer. The expression QTL approach should answer to your question. Thank you. But it would be very expensive and very difficult.
There's one question. There are two questions at the back. We'll take number one first. Then I think Francois has a question, and then we'll go back to the rear.
Thanks, Chair. I am Gopalakrishnan. My question is to the last three speakers. When they were looking into the route, have there been any effort to look into the canopy architecture? And if it has not been looked into, is there any chance to improve the drought tolerance level by changing the architecture, canopy architecture, in addition to changing the root dynamics as well?
Thanks, Gopal. That's a good question. What we look for is when, I don't know, you're talking about the chickpea as well, I think, and let me tell about the rice story. As I was saying to Professor Fisher when he was asking, there are a lot of other data that we have taken. It's very correct to say that if you do not take care of the canopy architecture, the root architecture doesn't have any relevance, actually. You're right in the sense. But then what we do is when we now look at the kind of segregating material, you have all sorts of variation that come in the leaf area and the leaf angles and then the droopiness and so on and so forth. And then in the morning, someone was also talking about the green leaf area index. And then when we look at all these indices and then we classify them, one thing that we can look at is a yield potential that increased, which I didn't present in my talk at least. I don't know, but Mahendra, he has some information in chickpea. But then when we look at the canopy architecture, which is quite similar to that of what we look at in IR64 or the recipient parent, and then, the canopy is comparable. The difference that we have is actually in the roots and the efficiency with which the carbon is assimilated for the unit amount of water transpired. and then the total biomass must increase. And if you keep the hardness indices constant, the yield increases is the concept that we look at when we now use the physiological traits for breeding to improve productivity under water limited conditions. Does that answer your question? And
You want to say anything about
Yeah, François Tardy. It's a question about root types matter and together with Xavier Drey. Okay, so root types matter. My question is whether this pattern you have shown is valid for one genotype or can be extremely different for different genotypes. Basically, if I understand properly what Xavier said, many things will depend on the ratio of the xylem connectivity versus the radial connectivity. And the radial connectivity depends a lot of aquaporins. And I can testify, because we did that with François Chaumont, that the amount of aquaporins in maize is tremendously variable between genotypes. So if the amount of aquaporins varies very much between between genotypes, then I would have thought that your pattern will change very much between genotypes, and this might be a trait per se. I don't know.
Oh, thank you, Tertio. I would like to start to say that actually the aim was not to compare many genotypes. The types of study we do is actually quite time consuming. But the main idea was to try to understand. So now in the literature, if we ask where maize take up water, actually we don't have a question. We don't have an answer to that question. People dig the soil, take the root lengths, root distribution, and try to say, okay, if the genotype with the longer root should be more drought tolerance, which many studies shows that actually it doesn't work. So the idea was mainly to understand where the root takes up water. Of course, this will change. So these studies I showed was in homogeneously wet soil. And we know that actually as the soil dries, this pattern will again change. But what we could show actually that in a mature plant start to be so in the literature people, they say that these crown roots actually they are mainly for holding the plant, which is not true. We found that actually they are the main location for the water uptake. We also show that actually, so for a breeding program, what I would suggest is that now we saw that all these laterals of the seminals actually they were not active after 2 to 3 weeks. So an effort can go to a direction of having plants with less laterals, because having plants with less laterals goes back to what Xavier was saying, with having plant with less cost of the carbon, because then you can have this energy for producing longer root, and this longer root can tap more water from the soil. The things for the equatorial.
We'll have one last question. Oh, sorry, you want to answer as well. Sorry.
Yes, there are two things I wanted to add to Mute's answer. The first is that the aquaporine question. So when we talk about the Kr over Kx, the Kr should in principle be extended to Kr plus the conductivity of the rhizosphere. So the things that occur in the rhizosphere would have exactly the same effect and need to be taken into account. And the local conductivity drop that Andrea was mentioning this morning, we can actually see this occurring in rhizotron with very high resolution cameras. The other point is we do not have to forget the complete system. So if one part of the root system or one root type is more conductive at the moment, then water will be preferentially taken up by this, and the rest would be just apparently not taking up water at that time, but might be the day after. So there is a highly dynamic.
If I could just comment on that, actually. We've done some split root experiments as well, because with our hypothesis that the tension in the system is going to affect the activity of the aquaporins. If you have some dry soil on one side for one part of the roots, it'll shift just because of the water potential gradients, more water flow to the other side. That corresponds with an upregulation of the hydraulic conductance in that side of the root system and an increase in expression of aquaporins. So it's incredibly dynamic. It's not just the gradients that matter, it's that's regulating the activity of aquaporins.
Just the point I wanted to make is before we see differences between genotypes,
we probably should be careful before telling any breeder you should select for that, because we take a big responsibility. I think we have to be quite clear.
I fully support.
Thanks very much, Francois, for that final point. And we will take the final question from the back. And please introduce yourself, because I can't see who it is.
Okay. This is Varaprasad from Kansas State University. My question is, across all of your speakers, you've talked about several root traits. You talked about rooting depth, root volume, root weight, and root numbers. Based on your experiences, which of these traits is important for effective capture of water? And number two, is there a relationship between any of these root traits with grain yield?
Who wants to answer that?
I think, Vara, it depends upon the target environment you want to look for. For example, in a terminal drought situation, you got rainfall coming nicely, then the top roots are going to feed the plant nutrients and water. As the season progresses, no rainfall, the top 30-40 centimeter layer dries up, then the deeper roots will be pumping water, especially if the soil has the capacity to store that water and water is available. In relation to grain yield, yes, we have already seen the work by Equisat in relation to that classical molecular work, breeding back crossing. Of course, if you go and look in the germplasm, you won't get that like any other traits. So based on the science, we heard the last couple of speakers and this today, we believe that root traits are important and we can manipulate as long as we know the fundamental, and we must target the environment.